All right, great. Yeah, 안녕하세요. Nice to meet you, everyone. Today, I'm sure I don't speak Korean, but I'm going to be talking to you about our spatial biology solutions this afternoon. So hopefully I'll try and keep it at a pace and everything. If there are any questions or if I need to slow down, please let me know. So I think it's important to talk about the context of why spatial biology is important. So the field of biology is certainly changing and evolving over time. And I think this technology is really groundbreaking, and there are new cutting edge tools being developed to address a lot of questions in tissues. So if you consider the evolution of genomics technologies, of course, back in 2009 was the launch of NGS sequencing and bulk expression profiling. And this is, a seminal moment in quite a few careers. I know NGS launched during my postdoc, and it's something that I worked on, and now it's a very common tool that's used. Of course, with bulk profiling, what we do with the tissue is that we take that bit of tissue and we homogenize it. So we don't really know what specific transcriptomic signal is coming out of what specific cell, right? So it's more of an average of the transcriptomic profile within the area that we're sampling from. So if we fast forward to 2012 and really the launch and the advent of single-cell RNA-seq technologies, this is a major breakthrough in technology where we were able to take a tissue and we were able to disassociate it into individual cells, and we're able to phenotypically profile them according to transcriptomic and even proteomic modalities now with single-cell RNA-seq. Of course, with tissue, cells really don't like to be disassociated. They like to stay in their native state. In the single cell RNA-seq, it's essentially a parts list. So we know what cells are present, but we don't know how they're arranged in space. And that's really something that's very important to understand biology. So if we fast forward to 2019 and the advent of the spatial biology, revolution, we launched our GeoMx DSP in 2019. Now what we're able to do is we're able to identify where these individual cell populations are arranged within the tissue. And we have the type of information, whole transcriptome, high-plex proteomic information of those cells as they're arranged in space. And now we can understand the interaction between those cells and how that leads to biological function in healthy tissues and also in diseased tissues as well. So to give you a practical example of that, this is a paper in the Journal of Clinical Investigation that we did in collaboration with Merck. And we were looking at the efficacy of a PD-1 checkpoint blockade therapy. So we know, it's extremely important with the advent of these IO therapies, but not all patients respond to that. So we wanted to understand why. So what we did in this study is we took some clinical tissue from patients, and we were able to give them Pembro, and we ran them through a bulk gene expression profiling modality on our nCounter platform. So we can profile up to 890 individual RNA species in a bulk way. And what we did is we identified an 18 gene signature that was predictive of patient response. And if we look at the graph down here on the bottom, on the x-axis here, we have that 18 gene signature score, and we call it the TIS score. And on the y-axis, is progression-free survival. So on the left-hand side, what we find is that the patients that had non-inflamed tumors rarely responded to checkpoint therapy. And then if we look on the right-hand side, the patients with inflamed tumors were the ones that were responsive. Now, however, when we look at this from a bulk approach, there was a subset of patients on the bottom right-hand side here. You can see that we're not responding to this checkpoint therapy blockade. And if we use bulk expression alone, we're not able to understand why, resolve why that is. So complex biology, or just biology in general, it requires to look into the tissue, so deeper insight into answering specific questions. So what we have here we go, okay, is we have two pieces of tissue from that study, right? On the left-hand side, we have a hot tumor, but excluded tumor. And on the right-hand side, we have a highly infiltrated tumor. Now, both of these have a very high 18-plex gene signature that we identified with the nCounter, as I mentioned before. So based on the bulk transcriptomic profiling, they would essentially look the same. However, when we look at the naked eye, we can see on the left-hand side in green here, we have the tumor. And then in the blue and magenta, we have some CD45 and CD3 labeled populations. We can see that the immune infiltrated stroma is, there's a high degree of number of immune cells, but they're not penetrating into the tumor. Whereas when we look over on the right side with the same markers here, we can see that those immune cells are now getting inside of the tumor. So based on this information and the bulk gene expression information, we can understand what patients may actually benefit from that PD-1 checkpoint blockade. And that's really the power of spatial biology. So we can leverage the power of bulk NGS plex, and we marry that with histology. And traditionally, up until, a few years ago, about 4 years ago, there's a paradigm shift that's occurring. So you had high-plex expression profiling. So this includes NGS, single-cell RNA-seq, flow cytometry. There's a high degree of plex, quantitation, and precision, but of course it lacks the spatial information that I mentioned before. Whereas when we look at histopathology and looking at imaging kind of data, it's typically low in plex. You retain spatial information, but it's semi-quantitative in nature and lacks precision. So what our platforms do, both the GeoMx and the SMI, is it marries all of those parameters together. So you have high plex, spatial information, quantitation, and precision within your tissues. So we know that the tissues that we work with, they're very precious. So whether or not these come from a clinical trial or if they come out of the lab, we're working on specific animals or something like that, these specimens are extremely important. So traditionally, to get a tremendous amount of information out of them, you'd have to take multiple sections of that tissue. So that can include for IHC, single-cell RNA-seq, bulk expression profiling, what have you, you would have to take multiple sections. But with our platforms, what you can do is you can just take a couple of sections and you can get high-plex multi-omic information directly out of that sample. So I know Philikria has spoken with you, but from Nanostring, who we are, just a little bit about who we are. We're a research-driven company. So we develop and we manufacture all of our own chemistries and platforms from the ground up. And that's really who we are. So a group of scientists that are working together. And on the right-hand side here, this is Dr. Joseph Beecham. He's our Chief Scientific Officer and Senior VP of R&D. And on the left-hand side here is Dr. Gordon Mills, who was a collaborator with Joe at the time. So Joe was at a conference at MD Anderson Cancer Center back in 2015. And he was having a conversation with Gordon, talking about how could he develop a chemistry that could perform unlimited plex in tissues for both protein and RNA. And during that meeting, he penned the original concept drawing for our DSP chemistry on a hotel memo pad. So this actually occurred at a conference. So it's pretty amazing. I think science in motion, really. Now, during that conversation, Gordon, he spoke to Joe and he said, there's two things that need to occur here. So one, this technology has to work in FFPE tissue, right? That's the treasure chest of information that comes out of the clinic and what's going to be the most viable. And two, the solution has to scale. You have to be able to ramp up production and get through quite a few samples. Joe took that to heart. So this is our ecosystem. So the first system that we developed was the nCounter system, as I've already mentioned. That was used in the Merck study that I just mentioned a few slides ago. And the nCounter system, it was designed to work in FFPE, very short probe sequences. We have zero enzymology, zero amplification, and we can perform direct molecule counting. Okay? So this is the original chemistry that we use, the foundational chemistry that we use to develop the GeoMx chemistry and also the CosMx chemistry. And the CosMx was launched in 2022, just in December of last year. So our new platform, the CosMx, as well as GeoMx, is gaining a lot of attention and accolades. So it was labeled one of the top 10 inventions of 2022 by the Scientist magazine. And our foundational chemistry paper and application paper reached the cover of Nature of Biotechnology in November of last year. So we're very proud of these things as we begin to innovate and find more discoveries. So the chemistry for both of these platforms is really predicated off of this in situ hybridization strategy, right? So we use very, very short probe sequences. And because of that, we're able to plex to extremely high level. That's one of the benefits of the GeoMx and the CosMx platform. So on the GeoMx side of things, we use a series of in situ hybridization probes for RNA detection. Now this is non-poly A capture. We're targeting along more conserved regions of that transcript. And on the protein side of things, we use a series of rabbit, mouse, or rat monoclonal labeled antibodies. And then in order to quantitate each of these analytes, we have a synthetic DNA barcode that we have covalently bound to either the antibody or the ISH probe via a UV photocleavable linker. Okay? And within that chemistry, what we're able to do is outline specific areas of biology, which I'll talk about in a little bit more detail later. But essentially what we're doing is we're cleaving these barcodes off of the ISH probe or the antibody, and we collect these and we perform an NGS readout. So this is a digital quantification of the protein or the RNA. On the CosMx side of things, we use a series of in situ hybridization probes and antibodies again. But in order to quantitate, we're using a series of cyclic hybridization and quantitation. So we're actually quantitating by imaging these particular species. And I'll talk about this in more detail a little bit later. If you'd like to do a deep dive into either of these chemistries, both of them were published in Nature Biotechnology. So very high degree of standards in terms of what we're publishing. And you can have a look at those at your leisure. coming back to Joe and Gordon's conversation that they had about scalability of the platforms, we've taken that to heart when we designed these from the ground up. So the GeoMx and the CosMx use industry standard tissue processing methodologies. So for both platforms, we just use a positively charged histopathology slide that you'll get out of the lab. So no proprietary slides. You affix your tissue to that slide, and then you can run those on either the GeoMx or the CosMx system. So if you've run an IHC, multiplex IF, or ISH experiment, you'll be able to run a CosMx or GeoMx workflow. And both of these, as I mentioned, the GeoMx is an NGS output, the CosMx is a standalone system, and both data sets towards the end of the year here will be able to be analyzed on our Atomics platform. So this is our cloud-based image and data analysis. So there's a high degree of flexibility with both platforms in terms of the types of studies that you can design. Because it's just a positively charged histoslide, we can use very large bits of tissue. We can use multiple tissues. So here we have some mouse brain examples. You can use core needle biopsies, mouse tissue, whatever it may be. It can fit within a very large scanning frame. So 20 by 15 millimeter for the CosMx system, or 35 by 14 millimeter for the GeoMx. Another very popular modality that we have or study design is with tissue microarrays. Okay. So not only can we increase the throughput of the number of samples that we can profile on an individual slide, but it also helps with reagent economy. So it drives down the cost of your experiment by using TMAs. This is extremely popular in Korea, by the way. Most of our studies come up on TMAs. So that is our ecosystem. So we have the Atomics, the GeoMx, and the CosMx. And in terms of what system should I select for my study really depends on your biological question. So with the GeoMx instruments, this is a multicellular approach. So what I mean by that is that we can look at a specific environment. We'll use the tumor microenvironment, for example, because I know this group works in oncology. So in that regard, we can look at the tumor itself. We can look at cell populations within the immune cell stroma. So T cells, for example, we can mask over those areas, and we can collect whole transcriptome information or up to 150 proteins. That's also going to be scaling as well. Right, so a high degree of plex, but it's not down to the single cell resolution. If you require individual single cell resolution or subcellular resolution, that's a question for our CosMx SMI system, where we can look at individual cell populations and we can look at the localization of RNA transcripts and protein species within the cell itself. So to give you a quick example of what that looks like, here we have a sagittal section of a mouse brain. So it's stained with alpha-synuclein, which is a key target in Alzheimer's disease. And what we've highlighted here is the hippocampus. Right? So on the left-hand side, we have a GeoMx profiling strategy where we're able to outline that specific structure with no gaps in the tissue whatsoever. And we're able to collect whole transcriptome information or high-plex proteomic information. And this is important for understanding biomarker discovery, looking at specific signaling pathways, and IDing particular markers that might be a response to a drug or predictive of a response, what have you. So there's a variety of applications there. Now, if you're interested in the individual cells that comprise this particular structure, then you would move over to the CosMx, and we can bifurcate individual cell populations and novel cell populations that exist. So I'm going to talk about the GeoMx a little bit here. So as I mentioned, we have whole transcriptome information. So we have targeted panels as well, but we can do whole transcriptome for human and for mice. And this is a biology-driven kind of approach. So as I mentioned, we can outline specific structures or compartments within the tissue. Any sample, so you can process FFPE, fresh frozen, whole mount TMAs, organoids or spheroids. As long as you can stick that tissue to the slide, we can profile it. This includes bone as well. We're a multi-omic platform, being able to analyze both RNA and protein, as I mentioned, and it's a scalable throughput. So the GeoMx is really the workhorse instrument that would be in your laboratory. We can get through quite a few samples at once, up to 60 samples at a given time. So I've already talked about the chemistry and that we're collecting these barcodes. I'm just going to skip through here really quick. What I will say as well is that we use two types of reagents on the system. So we use our profiling reagents, which is to quantitate RNA or protein, and then we also use imaging reagents. So this is to stain up to we have 4-channel microscope on the system, which is the FITC, Sci3, Sci5, and Texas Red channel. And if you can label your cells with an antibody that excites in any of those channels, then we can visualize them on the platform. So it's completely open. You can use clones that you're using in your laboratory. So we stain with both of those reagents, and then the slide is ready to go onto the GeoMx instrument. So I'll walk you through the workflow here. Okay. Again, so we're going to stain the tissue section with content for protein or RNA. You can also look at both species on the same tissue at the same time, which is very unique. We stain with the fluorescent labeled antibodies. So in this example, we might be staining the tumor. We might be looking at a macrophage population and maybe a T cell population, for example. So what we do is we take a high resolution multiplex IF image of that, and then we highlight what we call regions of interest, or ROIs, right? So an ROI is an area that we're interested in. This can be tumor microenvironment, for example. So what the system will then do is it will ID one of those regions of interest. It will shine UV light over that area, and then that will cleave the barcode off of the antibody or the ISH probe. Those barcodes are then collected and deposited into a single well of an on-instrument 96 well plate, and then that process is repeated for each individual ROI that we've highlighted, arranged on the tissue. Those barcodes are then collected. We go through a very simple NGS library prep. Those file types are then converted into one that's recognized by the GeoMx instrument, and then we have an analysis suite where we have a variety of tools that you can use to interact with your data and to generate specific graphs for your analysis. So I think the power really of the GeoMx instrument are these digital micromirror chips that are housed on the system. So there's 2 semiconductor chips. They're about the size of a human thumbnail, and they're comprised of millions of tiny little mirrors. And what that allows us to do is outline these non-contiguous regions of tissue. So we can outline unique biology and structures within the tissue down to single cell precision. So what that allows us to do, we have a colorectal cancer biopsy here. The tumor is stained in yellow, and then we have a pink stain for a CD45 population. What we can do then is, with those fluorescently labeled antibodies as a guide, we can create a tumor mask, which you can see on the top right-hand side. Everything highlighted in white, we shine UV light over, and then we collect whole transcriptome information directly out of that compartment. We can then flip that mask over to highlight the tumor microenvironment, and then we can collect information directly out of that as well. So what that means is, if we're looking at the ROI as a whole, this signal would be blended, but with the ability to segment between 2 different compartments, we get a better understanding of the biology at play within that particular area. Now, with any given AOI, you can segment up to 3 to 4 populations of cells within that area. So there's a lot of flexibility with the platform, as I've mentioned, with the ROI selection. So you can select geometric shapes. So these can be anywhere from 50 micron up to 660 micron in diameter for the circles. They can be squares, rectangles, or freehand polygons that you can draw around unique structures. The hypothalamus was one. You can draw around a glomerulus example, for example, in the kidney, whatever it may be. Within the geometric shape, you can apply the segmentation strategy. We can also do cell type specific masking. So the tissue does not need to be contiguous. The cells can be dispersed over the area. And we can also perform contour profiling. So you can place, equilateral tracks along the tissue. And in each of these tracks, this is an individual sampling event where we can look at whole transcriptome information as well. Or if you'd like to look at the entire tissue itself, you can do so. Here we have some examples in neurology as well. So we work in a variety of tissues. So I think we've worked in over 100 different tissue types since we started. So I just wanted to highlight this. This is something called our Spatial Organ Atlas. So what this is we've profiled healthy organs in human and in mice, and we've ran our whole transcriptome atlas to provide foundational data sets for our customers to download to use as a reference. And we also use it as a guide to show how we would draw regions of interest in specific tissues to show our customers how we would use our technology. It also provides guidance on the fluorescently labeled antibodies we would use to profile the kidney, the pancreas, the colon, et cetera, et cetera. This is an interactive software, and I highly recommend that you have a look at it. As a brief example here, what you can see, we're looking at the pancreas and specifically an islet of Langerhans. Right? So this is a 200 micron structure that we have here. We've used fluorescently labeled antibodies to isolate the alpha cells and the beta cells. And you can see that this structure, all of these cells are interdigitated. Right? This is a complex structure. With our segmentation strategy, what we can do is we can pull the alpha cells and the B cells apart from one another, and we can look at the entire transcriptome information out of those different areas. So as you can imagine, if we're looking at healthy tissues, we're looking at diseased tissues, we can see variations in signaling. If we're treating this particular patient with IO therapy or whatever drug it might be, we can also see the variation that has on the expression profiles, as well as different signaling pathways within that area. So a very, very powerful technology. As I mentioned, we have whole transcriptome assays. We have targeted assays, around 1,800 for cancer-specific profiling. And then our protein assays, we have content for immunology, oncology, and neuroscience. So the protein assays are built out in a modular fashion. So there's a central core module that's essential for the assay. And then you can pick a la carte content for specific targets of interest. And it's completely up to you. We can also customize the assays as well. So you can spike in up to 100 genes into the whole transcriptome atlas, or you can create a complete standalone 400-plex RNA assay for any species. All right, so if you're looking at infection studies or maybe HPV, and how that relates to tumor progression, that's something that we can look at as well. We also have the ability to look at RNA and protein on the same tissue. So again, up to whole transcriptome and 150 proteins on the same tissue. We are the only platform that can plex at that high level and do that on the same tissue. And we're working towards that on the CosMx as well. We have partnerships with Leica as well. So in terms of adopting this system into your lab, you can purchase a Leica Bond RX, and you can do all of the staining upstream, which reduces your hands-on time to only 30 minutes. And we've looked at slide to slide, user to user, and instrument to instrument performance when using this platform. This is what we use in our house, in our R&D labs internally, and the reproducibility is exceptional. We also have partnerships with Illumina as well. So using their Dragon IT pipeline, they have specific pipelines that are developed to convert our pipelines into file types that are recognized by the GeoMx instrument. So both of those solutions, what they're allowed to do is enable a quicker turnaround time and a faster time to discovery. I'm going to close out the GeoMx here with just a couple examples from the literature. So I think it's important to kind of come back to where we started. So the very first publication that we had on GeoMx was a protein study that we did with Jen Wargo at MD Anderson Cancer Center, and also James Allison, who is a Nobel Prize winning laureate. So in this study, we were looking at melanoma and the importance of what we found was tertiary lymphoid structures actually in the response to immunotherapy. So when we were going through this, it was interesting. We highlighted these B cell markers within the tissue, and Jim and Jen came back to us and they said, look, we should, I think it's wrong, your system's wrong. We shouldn't have some B cells in these tertiary lymphoid structures. But lo and behold, we went back to the raw images and we found out that that's actually true. And we discovered tertiary lymphoid structures. So this was not a field of study at the time. We enabled that discovery on our platform. So pretty exciting. 15 months later, Jen and her lab, they ran a follow-up study based on, some of those findings, and they, expanded on the role of these tertiary lymphoid structures in regulating immune responses to melanoma, and also looking at the heterogeneity of B cells and T cells within that environment. Funnily enough, at the same time, Goran Johnson at Lund University also ran a similar experiment, and they actually found the same exact result nearly at the same time. So both of them submitted these publications to Nature, and they talked to us, and they accepted both of these papers back to back in the same issue of Nature at the same time. So what an amazing orthogonal validation, of our platform and these findings from two independent labs that had not spoken to each other. We have more content that's coming out in the second half of this year. I know you're new to the platform, but we're having more sophisticated ways to draw these regions of interest to help automate that process for you. Some reagent changes. We'll be having an increase in protein plex on the GeoMx instrument, so surpassing that 150 plex. That will be announced at SITC this year. I'm sorry, not at AACR. And we also have a TCR diversity panel. So if you are interested in TCR diversity and profiling within a tissue context, we're the only company that's offering that right now in a spatial domain. Okay, so I'm going to shift gears over and I'll talk about our latest system. This is the CosMx Spatial Molecular Imager. A little out of here. So this is a raw image output from the CosMx SMI. So what you're seeing here, this is a 5-micron thick melanoma tissue at 1,000 plex RNA. And each of these individual dots that you can see here is an mRNA species that is resolved in space through a three-dimensional structure. So we can actually visualize where these transcripts are sitting within the cell. Now, for context, we can profile 1 million cells essentially on a single slide, and we can run 4 slides at once on our system. So that represents 4 million cells and 2 billion transcripts in a single SMI run, right? Now, the Human Cell Atlas project that was run over the past few years, right, They profiled 26 million cells. So what we can profile in a single run is 15% of that total number. Not only that, but we can also provide spatial context. Right? So more information than 406 labs generated during HCA. And that's really where the technology is going to. So it's a very exciting time. So the CosMx SMI, basic metrics for what we have the highest plex on the market. So we have a demonstrated 1,000 plex panel, and that is now moved to 6,000 plex, which is going to be available in Q1 of next year. As with the genomic system, you can profile FFPE, fresh frozen, TMA, and organoids. And it is multi-omic as well. In terms of all the in situ profilers that are out there, we are the only platform that offers RNA and protein quantification on the same system. And it's a tunable throughput, anywhere from 2 to 20 slides per week, depending on the assay that you choose, and a high degree of resolution as well. So we can get down to 50 nanometers in the X and Y direction. Now, even though the platform only recently launched, it is a tested and proven platform. So we have something called the Technology Access Program, which is a proof of concept lab that we run out of our R&D labs in Seattle. So we've had some customers from Korea that have used this service as well, quite extensively. But essentially what you do is you send us a couple of slides. We will take that. We will run an assay for you on the CosMx system. And in about 10 to 12 weeks, we will give you the raw data back so you can have an idea of how the system is actually performing with your tissue type. So we've delivered over 140 projects over the past year and a half. And because of the plex level that we're able to achieve, we've profiled over 70 plus tissues in both normal and diseased settings for both human and mice. So this is the chemistry. I'll explain it in a little bit more detail. So on the left-hand side, you have your native mRNA species. So we have target binding probes that are very, very small. And this is extremely important. So they're only 35 to 50 nucleotides in length. And we use 5 tiles per mRNA transcript. But we really only need one of those to bind in order to get a signal. As with the encounter system or everything else, we use 0 enzymology and 0 amplification of our signal. So that's extremely important when you're considering plex. We're able to plex to a much higher level than any other platform because of this design. Now, to the target binding domain, we have a readout domain, which you can see here. And if you can see each, there's some alternating blue spot areas. We call these spots. And we go through a process of cyclic hybridization and imaging. So what we will do is we will flood the tissue with these reporter probes. And then one of those events will have a reporter that will bind to one of those readout domains. We will then cleave that reporter off, and then we'll wash it and flood it with more reporters. And during the course of that 16 cycles, we will have 4 reporters that will bind to that readout domain, as you can see here. So here we have a reporter binding, we cleaved it, one of the spots is taken up, and so on and so forth until the very end of it, we have 4 of those spots that are occupied by a reporter. So, so that's how we do the chemistry, or that's the chemistry itself. So it essentially generates an optical barcode at the end of that cycle. On the protein detection side of things, we use a series of antibodies. We have a site-specific linker, of course, and then we have a readout domain, and then we go through cyclic rounds of hybridization and imaging with that. So for those of you that have worked on single-cell RNA-seq, we know that our endpoint of a single-cell RNA-seq experiment is the UMAP. Right? So we've identified individual cell populations according to their gene expression profile. But with the CosMx instrument, what we can do now is we can actually look at that UMAP, and we can look at where the individual cells are actually distributed within the tissue. Right? So we're adding now a Z or spatial component to that single-cell RNA-seq experiment. And this is how we do that. So again, this is looking at that melanoma sample. This is 1,000 plex RNA in a 5 micron thick FFPE image. And we take 8 to 9 Z stacks to the tissue, and we register that 1,000 plex RNA through each of those Z stacks. So what you can see here is this cartoon start to come up to the top here. So here in this opaque region, this is a nucleus of a T cell. And right here, we have the distribution of some selected RNA species within that cell. And you can see that we have a nice formation of those clusters around the cell itself. Right? And what we can do here is we can turn on and off our genes of interest. And if you can see, we can actually localize, specific nuclear genes, cytoplasmic genes, and ones that sit around the boundary itself. So that's one component of it. And then we also couple that with cell segmentation. So when you're considering an in situ platform, cell segmentation is absolutely critical. We need to make sure that we define the cellular boundaries through that three-dimensional structure so that we can accurately register the RNA or the protein species within the tissue appropriately. Without that, you're doing yourself a disservice, and you're not capturing the biology that matters. So at Nanostring, we are the only company that uses protein-guided and RNA-guided cell segmentation. So we have a specific algorithm that we use, which is called CellPose. It's an open source algorithm. And what we do is we stain the nucleus, we stain the cytoplasm, and some cellular boundary markers. And then we put that information and the RNA information into our algorithm to get an accurate definition of cell segmentation. What other companies will do is they will only stain the nucleus, and then they do a 15 micron expansion out from that cell, and they call that cell segmentation. They're not accurately defining the boundary. So it's a very important point to consider. Okay, I've talked how we've done that. So today you've heard me talk about plex a lot. So that's one of the benefits of nanostring technology. If that's one takeaway you have today, this will certainly be it. So when you're considering in situ profilers right now, the highest plex is 1000 plex that we have commercially available. And we'll be launching 6000 plex in the first part of next year. So why that matters is with other in situ platforms, I should mention, they only can go up to 500 plex. So why does that matter? So when you're considering cell typing or looking at specific cell states, right, you need a couple hundred genes in order to do that. Outside of that, you're not going to be able to look into additional bins of biology without increased plex. That's why it matters. Right, so the more plex you have, the more targets you can look at, the more biology and the more discovery you can make. So the types of applications that you can perform with the CosMx SMI. So typically the first port of call is looking at cell atlasing. What is my tissue? What are the cells within that tissue and where are they located? Right? That's the most important thing and usually the first port of call for most of our studies. Then we also look at cell neighborhood analysis. So neighborhoods are now looking at these cells and defining them into specific structures within the tissue. Right, so for example, if we're looking at the tumor microenvironment, what is the stromal component? What is the tumor component? How do those cell populations change in different patients if we're looking at different stages of cancer? If we're looking at different treatment groups, et cetera, et cetera. We can also look at the cell state and function of individual cells. You can look at that temporally or in any following particular treatments. Another very important aspect as well that our high-plex enables is the ligand receptor interaction. So we have 100 individual pairs within our 1000 plex panel, and we have over 400 in our 6K panel. So we can actually look at individual cells and how they're speaking to their nearest neighbor, right? That's extremely important. And then also, we're able to look at pathway analysis and biomarker discovery, looking at differential gene expression within the tissue. Now, this is not enabled right now. It's very complex because, again, you need to accurately define the cells within that three-dimensional structure to make sure that we can look at the conversations happening between each of those individual cells and what's differentially expressed in an accurate manner. In terms of content, we have our RNA assays. I've already mentioned we have 1000 plex panel for human. We also have one in mouse neuroscience. We also have a targeted 100 plex human IO panel. And we also have the ability to customize as well. So you can spike in 50 genes to our existing 1000 plex panels, or you can create a customized 350 plex panel that is standalone. We also have content for protein. So we have a 64 plex human immuno-oncology panel. And we also will be launching in Q4 of this year, just in a couple months, a 64 plex mouse neuroscience protein panel as well. We just learned about that today. I'll take you through some of those metrics. So here's spatial proteomics. Protein is extremely important, in tissues as well. So nothing defines structure within a tissue better than the actual protein content. So this is unique to what we can do. So with this 64 plex panel, you can see all of the different bins that we're looking at. So different markers for inflammation, checkpoints, T cell activation, myeloid components as well, different cell populations, and also looking at, potentially phosphoproteins and other key markers as well. So you can process one slide per day, and you can also customize. So you can add up to 8 additional targets into this panel. Right. So all of these panels, they have been validated. So we use cell pellet arrays to do that. So what we do is we take in this example for a protein, we look at 52 individual cell lines. And we do pre and post conjugation screening of our barcode to that within the tissue. We have a pathologist review each of those stains pre and post conjugation. And then as we integrate individual proteins into the mix, we look at the performance of those on their own and also in combination of the cocktail of antibodies that we use to comprise that particular panel. So as we've done this, we've looked at slide to slide, instrument to instrument, and looking at automated sample preparation, and we get nice reproducibility and concordance between our results. We go through a similar process for the RNA species as well. We use cell pellets for validation. Okay, so here's an example of some protein data. So this is from a clinical trial in breast cancer out of Oregon Health Services from Gordon Mills Lab. And what we're looking at here is a core needle biopsy. And in this little vignette, we're looking at an area of the tissue. And what we're looking for is the phosphorylation of ERK1 and 2, right, into the nucleus here. So this is extremely important when deciding if patients are good candidates for tyrosine kinase inhibitors. So this one was a phosphorylation inhibitor, was provided with a phosphorylation inhibitor. They took a sample 2 weeks following this, and they found that there was a complete decrease in MAPK and ERK signaling following treatment with that inhibitor therapy. Right, so this is how it's being deployed within the clinic, at least in one example. In that same study as well, so they're looking at a combination of RNA and protein within the same species. So on the top left-hand side here, we can see that we have a core needle biopsy. And on the bottom left, we're looking at the phenotyping of these particular cells according to specific antibody markers for CD3, CD8, CD68 for some conical macrophage markers as well. And then we looked at a 1000-plex RNA panel. And also this is actually a 100-plex protein panel that we did through R&D. So if we look at the RNA side, of course, what we can see is that we have a tumor nest here in green. We have a T cell nest and some tumor-associated macrophages that we've identified within the tissue itself. So within the tumor-associated macrophages, we're seeing an expression of CD68, CD163. So that's a nice sanity check, and some markers for our T cells over on the right-hand side. And then when we look at the actual protein expression of those markers, we're seeing that it marries up with those expression levels. So again, we're having orthogonal validation of the RNA with the protein on the same platform. You don't need to vacillate between two different systems. Now, as we do a deep dive into more of the biology that's at play, like here we have highlighted the tumor nest itself, and what we can find is that there's a high degree of expression of macrophage inhibitory factor. And when we look at the macrophage population around the tumor nest itself, we can see that these macrophages are indeed not infiltrating into that space. Right, so that might be a guidance for therapy for that particular patient. This is an example that I'd like to bring up. So this is a non-small cell lung carcinoma study that we ran. This is one of the first studies that we released on one of our prototype instruments. So this is in the literature. And we looked at 5 individual patients. What I want to highlight here, which is important, is that we don't do any RIN scoring or DV200 scoring for sample quality on either of our platforms. It's not concordant with the performance. But we did it here to prove a point. So the RIN values for these patient samples were 2.4 down to unmeasurable, right? And DV200 scores from 64% down to 20%. So you couldn't even perform NGS on several of these. Right. So despite that, 94 to 98% of those actually passed QC on the SMI. So what that means is because of our short probe sequences and our chemistry design, we're able to work with very degraded species and very degraded samples, which is likely what you're to encounter from a clinic. Okay. So several groups have used that data set to publish a variety of different publications, Nature among them, which you can peruse at your discretion. We've also released another publicly available data set in liver cancer. So we have, what we've showed in healthy liver and in liver cancer, we have a high degree of sensitivity. So several metrics looking at the number of transcripts detected per cell, a large degree of diversity. So over 70% of targeted genes were detected across the tissue within that 1000-plex panel, and also a high degree of sensitivity. So we use negative probes and false code sequences within to determine detection, and over 99% of those genes were on target. And then, of course, I've already talked to you about our best-in-class cell segmentation, why that's important. Okay, I'm just going to talk about our 6K panel. So this we will be launching through our technology access program in Q4 of this year. This will be a commercially available product in Q1 of 2024, so just at the start of next year. So very, very exciting. We're able to measure over 400 ligand receptor pairs, and we're able to incorporate a customization of up to 200 additional RNA targets into this panel as well. So why plex matters, I guess, historically, I also want to highlight this. So AGBT is a, big genomics conference in the US that Nanostring has been attending for eons. But back in 2019, we launched the GeoMx. We were at 84 plex protein or RNA at that time, which is 20 times more than anyone else. A year later, we were at 1800. A year later, we were at whole transcriptome for GeoMx. Last year, for our CosMx assay, we showcased our 1K panel, which is shipping and available now. At AGBT this year, we showed 6K genes as well, which again will be commercially available in the start of next year. And Joe Beecham, our scientific officer, believes that we'll be able to get up to whole transcriptome next year. I won't make any promises, but I know that in 5 years, Joe hasn't missed the deadline. And if he thinks that we can do it, I truly believe that we can. So just to illustrate a point of why Plex matters. So if you're considering other in situ platforms, so this is looking at another company looking at their 248 Plex panel. In this Vernoid plot, everything in green is the biology that is covered by this 248 Plex panel. Everything in white is not covered. So this is the information that you get out of a very low Plex panel, which is where our competitors are sitting. This is the 1000 Plex panel. Right, so again, we've profiled this in over 70 plus tissue types. We've delivered over 130 projects, and we have, I think, over 20 now preprints and publications. This is actually a little bit older. So this is shifting over to 6K, right? So this essentially covers the entire human reactome within that tissue. So almost everything that you could want to try and find, you can with our 6K panel. So in Q1 of next year, what this is going to represent is a 6-time increase in our Plex capabilities, 12 times more than our nearest competitor. We'll also be looking at increasing the protein Plex in the latter half of 2024 or early 2025. And we'll have more content available as well. So we'll have content for mouse universal panels at the end of this year and the mouse neuro panel and customization, as I mentioned. So just briefly, I'll spend the next 5 to 10 minutes here talking about some of the 6K panels. So this is William Huang. He's a scientist that we've been working with at the very beginning. And in August of last year, he published in Nature Genetics on the cover his genomic study looking at pancreatic cancer using the genomic study. So we worked with Will. We gave him early access to the 6K data to run on those same samples. And this is some of the raw data that we found in that study. So we're still working through the analysis. But what we did show is that we can run that 6K panel. What you can see here is the raw image of that. So, as we scaled from 1K to 6K, the system was able to resolve the increase in Plex. You can see some of the raw images there. And we're also able to look at protein and RNA on the same slide. So of course we can do that, at 6,000 Plex to 8 Plex. We'd like to increase that as well, but here you can see some of the images. And again, the data from protein is extremely important when we're looking at receptor ligand interactions or just the structure of that tissue. So I think one of the most important things as well is in terms of looking at the panel itself is in real samples. What we can see is once we reach about 50,000 cells, we're capturing about 4,800 to 5,300 unique genes within that panel, right? So that's more than you'll find in a single cell RNA-seq. And we're keeping spatial context, and we're discovering more genes. And that's about 3 logs, 2 to 3 logs over our negative control and false probes, right? So we've shown that it can work. This is looking at the cell type distribution of that 6K Plex data. So we have, our different, we have some calves, dendritic cells, immune cell populations, et cetera. And at 6K, you can see the UMAP over on the right. We get a nice separation of these different clustering, right? So the more data points we have, the more genes that we have, the better separation and identification of unique cell populations within the tissue that we're able to perform. So this here, I think we were looking at about 5 or 6, 5 individual tumors, and we can see the separation of those within the system itself. So if we consider the analysis, so what we can do here, just going on to this Vernoid plot, you can ID, an area that you're looking at. So in this example, we're looking at signal transduction. So we can pick that and we can do a deep dive into that analysis. So here we're looking at 407 individual signaling ligands. And we went through and one of our bioinformaticians developed this algorithm to look at correlation matrices within the tissue itself. His name is Patrick Donaher. He's done a lot of brilliant work for us in the past as well. But as we start to look at these ligands and as they line up within the tissue, we are able to ID these specific hotspots within the tissue, particularly looking at signal transduction. So looking at chemokine and cytokine activators. So we looked at one of these clusters, and here on the right-hand side, you can see we're going to do a little bit of a deep dive into that. So if we look at this particular space, what we can see is that the conversations that are happening in the tissue vary between different cell populations. So here we have cancer-associated fibroblasts and macrophages together. We have CAFs, macrophages, and tumor cells in a particular cluster. And then down here, we also have macrophages, nerve cells, and malignant cells that are speaking to one another within a particular radius, right? So, trying to understand that biology, Will is working on understanding the actual biological impact of that. But this is the first time, as far as we know, that we've been able to actually resolve that type of data in space. So we haven't stopped there. We've also moved on to look at this 6K panel within the human brain. And what we've done here, this is, I think, in association with the Allen Institute in the US, if I'm not mistaken. But we're looking at these different correlation matrices throughout the tissue as well. And when we do that, on the left-hand side here, you can see that these are the signal ligand cascades between all of the individual cells within the human brain that we're IDing here. What we did notice was there was a single cell population that had 3 markers that were separated from the rest of the cells within the tissue itself. And they were expressing SST, NPY, and CORT. And we took that over to the Allen Brain Institute and we asked them what this is because we didn't know what we were looking at. And they said, oh, that's a very rare cortical interneuron called SST caudal neurons. And they're very, very difficult to find within the tissue spatially, but we were able to do that with our 6K. And you can see that we have those particular caudal neurons identified here, and you can see those 3 particular markers around there. So pretty exciting stuff, right? And this is just the tip of the iceberg. We're also looking at, as I mentioned before, looking at the 6,000 plex panel and increasing the plex on the CosMx from the 64 plex to up to 120. So what this allows us to do is really help, again, with that cell segmentation strategy and really define these unique cell populations that we find within the tissue. Here, we're highlighting a macrophage. And with those 120 plex proteins, you can see that we can actually highlight and look at the different projections that are coming out of these very complex cell populations, right? Cell segmentation is extremely important. And what that's going to enable us to do, and here, multiple examples of these microglia within the tissue as well, is to be able to resolve those, to look at them in a more informative way, and to be able to improve our cell segmentation. Last 2 slides here, I want to talk about our AtoMx platform. So this is the environment where you will be interacting with your data, both GeoMx and CosMx. And so this is a cloud-based solution. And what it allows us to do is perform all of our comprehensive data analysis within the cloud. Right? All of the information that you have out of these in situ platforms develops a lot of data or produces a lot of data, and that data needs to be stored somewhere. And we have a scalable compute and storage solution that's enabled by AtoMx, right? It represents about, it is based on a subscription, and the cost of that is about 10% of your reagent spend to house those projects within the cloud. It enables easy sharing and collaboration. So for these large data sets, if you're working with a pathology group or an academic group, or maybe another remote site of your company, you can email them a link, and then they can be embedded in that data, and then they can either run their own analysis or just have a look at the work that you've done and make comments in real time, right? And that's all enabled through the cloud. There's a high degree of security and compliance that follows all of the AWS structures, and I've already talked about the cost effectiveness. Another important aspect of this software development is our commitment to providing open source code and analytics for our customers. So you'll be able to customize and bring in modules as well. And we'll also be trying to work with other software companies as well, so to integrate third-party tools. And we're already starting to do that in the digital pathology space, and also working with some other spatial AI and machine learning companies to develop more tools and analysis modules for our customers. So what AtoMx does do, and not only is it an interactive image and data viewer, so you can look at the tissue and the actual outputs of transcript and protein data in real time, but it allows customization as well. So if you have a bioinformatics group that is developing pipelines in R or Seurat, they can download those into our system, and they can incorporate that as a module into the AtoMx pipeline. So it's an on-demand compute, as I mentioned, and it's also a site for collaboration as well. And we can also perform long-term storage, which you can access anywhere at any time. Now, one of the major barriers that does come up with spatial biology, just in general, is the ability to analyze that data and how do I interpret that. So it is quite a task. It takes a village to understand how to do that. But at Nanostring, we've developed a service called the Spatial Data Analysis Service. So this is really, a journey where we can provide you with your data, and then we can also give you an answer at the end of it. So this is a fee for service. But what we do essentially is we, obtain your data, so it can be generated on your own platform or whatever it may be. You pass that information to us. We'll perform QC and normalization on that. We have a consultation with you in terms of the hypothesis and questions and aims. What are you trying to discover within this particular study? We take that away and we develop the pipelines for you, and we come back in 3 weeks and we give you all of the source code and all of the information and the standardized report for the project as we defined together. And then we will walk you through that report and we'll provide you with the code that show you how to do your analysis. So pretty amazing, pretty powerful study itself, or service, itself. And this is something that we can offer you and help you along your journey. So that is the last slide that I have. I think I'm just on time. Thank you so much for your time.